welcome back to Biology 121 Online. This is video 5, Introduction to Genetics, this time with a mouse cursor to point at things. So we're going to talk about genetic inheritance today, and I want to make 6 points as we start. First of all, when I talk about inheritance or heredity or heritability, these are all very closely related concepts, and I'm going to be using them more or less interchangeably. So when we talk about genetic inheritance or heritability, there are 6 points that I want to start with. First, parents pass their genes on to their children. Genes may pass through many generations. The things that are inherited genetically can include physical traits, blood types, certain kinds of behaviors, and risks of disease. Dominant traits are more likely than recessive traits to be seen in offspring. Traits will often skip a generation, and in very few cases is this simple enough for a 9th grader to understand. So we'll break each of these 6 points down just briefly in this introduction. So first of all, parents pass genes on to their children. So when we talk about parents and children, we really are talking about sexual reproduction. Sexual reproduction occurs not only in almost all animals, but also in many, many kinds of plants and fungi. So all kinds of eukaryotes include sexually reproductive parent-children relationships. In cases of sexual reproduction, each parent contributes half of its genome. Now, a genome, this word here, is the entire set of all of the DNA in an individual. So when we talk about a genome, we're talking about a single individual's complete DNA, all of the DNA that individual has. So every parent contributes half of its total DNA to their children. So which half gets passed on to a given offspring is basically random. Any reproductive cell, also known as a gamete, is incomplete. A gamete cannot become a living organism all on its own. In humans, gametes include egg cells and sperm cells. But in plants, we talk about pollen, and in other kinds, and in fungus, we have spores. So there's different kinds of gametes depending on what kind of sexually reproductive organisms we're talking about. I'm going to make a lot of references to sperm and eggs, very likely, because that's what we kind of best understand. But understand also that when we talk about sperm and eggs, we're really talking more broadly about gametes also in plants and in fungi. So gametes don't become individuals of themselves. They have to fertilize with a compatible partner gamete. So in the case of animals, that would be a sperm and an egg. The process by which those chromosome pairs are split into gametes is a process called meiosis. Now, meiosis you probably did learn about as a 9th grade biology student, and what meiosis does is take the entire genome of a cell, split it in half, and put half of the genome in one gamete cell and half of the genome in another gamete cell. But it's important to note that those two gamete cells, each of which are what we call haploid, so they have half of the entire genome in them, are going to be the same kind of gamete. If you are a reproductive male, then your DNA gets split between 2 sperm cells. If you are a reproductive female, then your DNA gets split between 2 egg cells. So I want to make that point clear. Third point, genes can pass down through many generations. So consider it this way: every parent's DNA is itself made up of a 50-50 split of their parent's DNA. So on average, 1/4 of your DNA came from each of your grandparents. So in that sense, the DNA is being passed sequentially from generation to generation to generation. And as the second point points out, this DNA, the sequence of DNA, can be passed unchanged from parent to child to grandchild to great-grandchild to great-great-grandchild, and on and on and on. I just want to jump back and just highlight this word here. A particular sequence of DNA is in general called a DNA allele. So when we think about a sequence of DNA, this is the G's, A's, T's, and C's, and the order in which they come. And so an allele refers to any particular sequence of DNA. Now, our best estimate is that humans have about 20,000 different genes. And what a gene is, is a particular segment of your DNA that does a particular thing. So, for example, we might say, I'll refer, for example, to the BRCA1 gene, which is sometimes called the BRCA1 gene. The BRCA1 gene is a sequence of DNA that produces a particular protein that has a protective effect that helps eliminate breast cancer when cancerous cells arise in mammary glands. So different people have different versions, different sequences, different alleles of the BRCA1 gene. So the sequence, the particular sequence is called an allele, but the functional positioning of that DNA within your genome is a gene. Now, the alleles only change when a random mutation occurs, and that's why for most people, you'll most often have unchanged DNA sequences being passed from one generation to the next, to the next, to the next. However, because again, as you go further back in generations, your likelihood of inheriting a particular allele from a particular ancestor becomes increasingly small because the number of ancestors who are contributing to your DNA gets larger and larger and larger. Next point, we can inherit physical traits, things like blood types. In some cases, some certain kinds of behaviors are inheritable, and risks of disease can also be inherited. But really, the only thing that an offspring actually inherits from its parents is the DNA. If we think of the fertilized zygote, where, say in humans, an egg and a sperm cell have just met, we know that that one single cell, after dividing and dividing and dividing and dividing, is going to eventually, potentially, grow and develop until the zygote becomes a fetus, and the fetus, if born, becomes potentially then a living human, right? But the only part of you that you originally inherited was that one gamete from each parent. So you're only actually getting the DNA. That DNA encodes things like blood type, certain kinds of taste buds. You may have heard that some people taste broccoli as being very bitter and other people do not. That's actually something that is known. We actually understand what specific allele in which specific gene controls whether or not you taste the bitterness in broccoli. You might have an increased likelihood of developing certain diseases. So there are genetic proclivities that can be associated with these things. And the mechanism, the way in which DNA establishes things like blood type and disease risk, and in some cases even certain kinds of behaviors, is the particular variant of a protein that it encodes. So we know that different people may have different alleles or sequences of DNA, and those different sequences among different people produce slightly different variants of a protein. DNA is a set of instructions for how to make a living body. It's like a recipe book for making proteins. You may have a different recipe in your family for how to make chocolate cake than the kind of chocolate cake that my family makes. We're both making chocolate cake, but we're using a slightly different sequence of ingredients and actions in order to make our different chocolate cakes. That's the metaphor I like to use for how variation in DNA can result in different variants of the same kind of protein. Now, let's talk about dominant and recessive traits. First of all, dominant and recessive alleles are equally likely to be inherited. I want to make that really clear. But dominant alleles are more likely to be expressed. That is to say, they're more likely to become visible. And there's two different mechanisms that can help explain most of that difference in dominant allele expression. A recessive allele may be less likely to be read and used than a dominant allele. In other words, if you have two different recipes for the same protein, one of those recipes may sit on the shelf the whole time, while the other recipe is the one you're always using. And a recessive allele may also produce a less functional protein. So if your DNA doesn't actually have a recipe for chocolate cake, you're not making chocolate cake. And so that, if one person has one good recipe for chocolate cake and one recipe that doesn't even actually make a cake, then only the dominant one would be expressed, be visible. Finally, and this second, penultimately, some traits can skip a generation. So because chromosomes come in matched pairs, humans have 46 chromosomes in 23 matched pairs. So when you think about the genetic testing company 23andMe, well, the 23 refers to the what we call diploid, the matched pairs of chromosomes. But again, those come in doubled sets. So you always have two different copies for every piece of DNA in your genome. One of those copies you'll have gotten from one parent, the other copy you got from the other parent. If you have a mix of dominant and recessive DNA sequences, you'll express the dominant trait, but your offspring might inherit your recessive copy that you never used. And if a child receives recessive alleles from both parents, then they'll express the recessive trait as well. That's the really simple, like, Punnett square version of this that you learned back in 9th grade. But I gotta warn you, it's almost never as simple as what you learned in 9th grade. The story of Gregor Mendel breeding pea shoots is a very deliberate simplification of genetic inheritance. Even in his own results, he acknowledged that that was a deliberate simplification of what he really saw happening. So if a gene is, one of the reasons why it's never that simple, or almost never that simple, is that if a gene is really important, you probably have multiple copies of it. So these different copies, this is a feature that is sometimes called polygenism. These different copies of a gene may be located on different chromosomes entirely. So the different positions within your DNA on those 23 different chromosomes that the different copies of potentially different alleles for genes that make the same kind of protein are called gene loci. And loci is just a Latin word for the plural of location, right? So if you think of the word location and you shorten that down to loci, then you're talking about the locations on which these different genes might reside. So that polygenism is going to be a really important part of understanding why it's almost never this simple, and why, for example, trait skipping a generation is much more complicated than what you probably learned in 9th grade. Dominant and recessive traits is probably much more complicated than what you learned in 9th grade. And how we inherit things, especially things like about risks for diseases or behaviors, are much, much more complicated than how you learned in 9th grade. So we're going to leave it the introduction at that point. I really am hopeful that you're going to have lots of really, really good questions to raise coming off of this discussion, and I'll leave you with just one. How does DNA and genetic inheritance explain the entire range of skin colors in humans?